Hi, welcome to this lecture on environmental adaptation for animals. Now this video will focus only on one thing: how to bind the oxygen on hemoglobin or other carrier molecule and how to release it when we are using the oxygen for our daily use. Okay, the binding of oxygen depends on two things. First of all, the oxygen sensitivity or Hi, welcome to this lecture on environmental adaptation for animals. Now this video will focus only on one thing: how to bind oxygen on the hemoglobin or other carrier molecule and how to release it for the use of the cells. We have learned that the oxygen binding depends on two different things. So what is the oxygen affinity? So how eagerly the oxygen is binding on the binding sites. And then how much oxygen is available. So if there's no oxygen available, so if the partial pressure of oxygen is zero, then of course all these binding sites are unoccupied. There's no oxygen. And if there's enough oxygen or very high oxygen concentration, very high partial pressure of oxygen, then all these binding sites are occupied. And between them there's a sigmoidic curve, as well as there's a sigmoidic curve on how much oxygen we have in the circulation. So the oxygen can be binded in hemoglobin or it can be dissolved in the blood, but this dissolved oxygen is very minimal to compare the oxygen that is carried with the hemoglobin. And in high partial pressure of oxygen, all the hemoglobin is saturated, so that it cannot carry more oxygen with the hemoglobin at all. So that's why we can't carry in the blood more than 200 milliliters per liter of blood. And actually that's exactly the same amount that we are carrying in the blood that is leaving the alveoles. So that's why we have just this oxygen affinity in our hemoglobin. Okay, then we have oxygen for the rest of the body, and we had to release, and after that we had to use it. Or actually we started from the use. So we have mitochondria, and because the mitochondria are using the oxygen, so in that way, that's reason in the rest of the body the partial pressure of oxygen is lower. And in this figure it means that okay, we had to go on the leftward. On the partial pressure is smaller, it means that okay, because this curve is going down, there's less oxygen that can be binded in the hemoglobin. So it must release the oxygen. And this causes that in the resting cells or resting animal, we have used maybe 1/4 of the oxygen that we are carrying. So partial pressure is somewhere between 4 and 5, and there's the concentration of oxygen is now in the large veins, maybe 50 milliliters per 100 milliliters. So the blood oxygen utilization coefficient is about 1/4. But it can be enhanced when during the exercise we are using 3/4 of the oxygen in the circulation. Okay, how we can deliver the oxygen during the exercises? The problem is that okay, when we are exercising a lot, there might be a problem that okay, in the alveoles during the heavy exercises there is a little bit less oxygen available. But the good point is that because this is a sigmoidic curve, we are on the plateau. So even the small changes in the partial pressure, they are only minimally affecting on the oxygen concentration in the blood. So it's still quite much all the binding sites can be occupied even when there's less oxygen, so when the partial pressure is decreased. But then near the skeletal muscles, etc., where the partial pressure is lower, then even small drop in the partial pressure are releasing a lot of oxygen. And that's why we can carry more oxygen for the cells that are using a lot of oxygen. And furthermore, when we are exercising, we are increasing the blood flow, and that causes that, okay, we can get more hemoglobin and more oxygen for these tissues. And it's still quite safe to use even 70% or 80% of the oxygen, because actually 90% use can be released before the oxygen is on the partial pressure is under the critical point where the aerobic catabolism cannot be anymore working. Okay, but the hemoglobin has more than one binding site, so there was 4 oxygen that were occupied, or it can be also unoccupied. So what happens? In the previous video, we had a very simple equation that we didn't much care. It's called Michael-Mentor equation, and it describes a situation where we have a substrate where one ligand is binding, and this can be expanded to have several identical binding sites, like 4 binding sites of oxygen, and it's called Hill equation. And when you're comparing these two equations, the only difference exists that there is this exponential term h called Hill coefficient, and it means that how many binding sites there exist and do they cooperate. And when having a dissociation constant, for example 1, you can see that with this Hill equation we have a steeper S-shaped curve that is having, and then meaning that, okay, in low concentrations of the ligand, if it works with Michael-Mentor equation with one binding site, it will still try to keep it. But with this, if we have more binding sites, we release it. And the same difference exists between a monomeric globin and tetrameric globin. So human myoglobin is a monomer, binding only one molecule of oxygen. And that's why there's hyperbolic relation between the saturation and partial pressure. But in the hemoglobin, we have a sigmoidic relation, because more than one binding sites exist. And furthermore, these binding sites actually are facilitating each other. So when we have one more oxygen binded, it will help the other oxygens to bind. And in practice, this means that even in low partial pressure, the myoglobin is saturated. And practically this means that if we have low partial pressure near the skeletal muscles, the myoglobin will steal the oxygen from the hemoglobin. And most of animals have several binding sites. Over here we have vertebrates and invertebrates, and with different kind of oxygen carrying mechanisms, and all these have sigmoidic curve, meaning that, okay, there's more than one oxygen molecule binded on these respiratory pigments. But you can see that not only they are on different scale on the how much oxygen you can carry in the circulation, but also some are more on leftward and some are more on rightward. So that's why the partial pressure of oxygen that is needed for half saturation varies from 3, for example in carp, to 50 in octopus. And that means that the respiration pigments have quite variability in the oxygen affinity. And note also that, okay, one of the best in carrying oxygen in the blood is the seal, because it has these long divings. Okay, oxygen affinity is affecting that, okay, how much oxygen we have where it's saturated. And its easiest way is to check that, okay, on which partial pressure we get 50% saturation when half of the oxygen binding sites are occupied. And if the partial pressure is low, then we are calling that, okay, it has high affinity. So meaning that, okay, now in that case, this curve would be on leftward. But if the curve is lifted on rightward, we have low affinity. So higher higher partial pressure of oxygen is needed to oxygen binding. So different kind of partial affinities are having then different kind of partial pressure that is needed for 50% saturation. And shift to right then means that the partial pressure needed to saturate is higher, and the pressure that is needed for half of the binding sites occupied is higher, and that causes that, okay, then the oxygen affinity is lower. Okay, it should be quite simple, but unfortunately it's not. We have a Bohr effect. It's, first of all, it's caused by acidification. So it's called fixed acid Bohr effect. So if we have low pH, even in human hemoglobin, the affinity is lower. So this acidity is actually a competitive allosteric inhibitor. So it affects somewhere on the hemoglobin and reducing the oxygen binding. And therefore, at low pH, the hemoglobin has lower oxygen affinity and the saturated at higher partial pressure. But at the same time, it means that if we have, for example, skeletal muscles that are releasing lactate and lowering the pH, in that case, more oxygen is released on that area. But it's also affected on the carbon dioxide. So that's why we have also carbon dioxide Bohr effect. So beside the carbon dioxide it's affecting on pH, it's also affecting directly. And surprise, surprise, it depends on the species. So in some species, this magnitude is very large, and in some cases it's quite minimal. So in some cases, that means that the accumulate carbon dioxide in the circulation affects more drastically on some animals. And the good point is that, okay, when you have skeletal muscle that is using a lot of oxygen and therefore producing a lot of carbon dioxide, you are releasing at that area more oxygen. And this actually then means that now the oxygen affinity is modulated during the circulation, even with the same molecule. And that makes sense now that, okay, you can have higher binding in the alveoles, and then you can release the oxygen with the same molecule because now it has lower affinity. It must release oxygen to the surrounding. So to have some kind of summary that, okay, what is the real effect on the Bohr effect on the oxygen carrying and oxygen release, we can compare the arterial blood where the affinity is high to blood in the veins where the affinity is low because there is more carbon dioxide, there is lower pH, and then we can estimate that how much is the partial pressure and compare that, okay, if we would have the same affinity as we have in the atrial blood, we could release 3 milliliters. And if we could have the same oxygen affinity as we have in the venous, it would be 6 milliliter release of oxygen. But together, we have now released 7 milliliters. So we are releasing more oxygen because of this Born effect. And when comparing that what we could release if the affinity would be the same all the time. Okay, beside the Bohr effect, we have a root effect. But it's good that, okay, it only affects in some animals. So that's why it's easier to forget. And in vertebrates, it's been shown only on teleost fish. And this causes that not only the sensitivity or the binding affinity for oxygen is modulated by pH and carbon dioxide, but also the total amount of oxygen that can be carried. So the total amount of oxygen that can be binded is affected. And this affects then that when the animal is having low pH and certain tissues, it releases more oxygen. And that is the way how these animals, these fish, are filling the swim bladder. And they are carrying the supply of oxygen for the eyes. But then, oxygen is also affected by the temperature. So, when we are increasing the temperature, we are reducing the oxygen affinity. And this helps the release of oxygen during the exercise when we are having hyperthermia. But then, we had the problem if we have hypothermic limbs, so when we are reducing the temperature, there might be a problem to get enough oxygen there. Or the oxygen is circulating, but it's not released. And it can be also a problem for these cold acclimated ectotherms. When the whole body temperature is low, then the oxygen is just circulating. So still one more way to put it. So when having normal affinity with loading and unloading, and then reduced affinity, we can affect quite drastically, even with the same molecule, even when we are not changing it during the circulation. Because in the low affinity, we can release more oxygen, but still the saturation happens quite much in the same partial pressure. And that's why we can then release more oxygen for the circulation and for this use of the cells. Thank you.